Hi, I'm Soren Andrews, and today I'll be talking about the complexities of DNA replication. If you ever took biology in high school, you probably know that DNA is made up of many nucleotides bonded together in the form of a double helix. You probably also know that DNA replicates during the S phase of the cell cycle. Another piece of basic information is the help of enzymes to speed up replication. Some of the most common enzymes are helicase, primase, DNA polymerase III, DNA polymerase I, and ligase. DNA polymerase III is one of the most interesting and complex enzymes. DNA polymerase III adds nucleotides to each of the original strands. But there's a catch. It can only work in the 5' to 3' direction. To further understand what 5' to 3' means, we need to take a closer look at the molecular structure of DNA's ribose component. We can see there are 5 carbon atoms and 1 oxygen atom in the ribose component. We classify the direction of the strand by numbering the carbon atoms around the oxygen atom in a clockwise rotation, thus 1', 2', 3', 4', and 5' carbons. On the outside of each strand, the carbons will either be numbered 3', 4' to 5', or, in this case, 5', 4' to 3'. That's where we get the 3' to 5' directions, or the 5' to 3' directions. Since DNA polymerase III can only add nucleotides in the 5' to 3' direction, one strand will be a continuous strand of added DNA, whereas the other strand will be small fragments of added DNA. The continuous strand is called the leading strand, whereas the strand of added fragments is called the lagging strand because it takes longer. Each fragment is also called an Okazaki fragment, named after the Japanese scientist who discovered them. One major problem with DNA replication, though, is time. How can our genome of nearly 3 billion base pairs replicate itself in a short amount of time? Well, it's actually quite simple. Our DNA starts replicating at hundreds of thousands of different points along our genome. The interesting part is not all of these points of replication start at the same time. Their start time is dependent on how tightly the DNA or chromatin is packed. The less tightly packed sections of DNA will thus begin replicating before the more tightly packed sections of DNA. You can think of this process as fireworks lighting up the sky, bursts here and there specifically timed and coordinated.